I'd like to invite Dr. Micol Damiani of Imperial College London to present the work she did in her PhD with us on multiplex detection of biomarkers for liver cancer using a novel nanopore-based sensing platform. Welcome, Micol.
Okay, hello everyone, and thank you for being here. Today I'm going to present the work that I developed during my PhD, which was partially funded by the AMMF. So, as you all know, a lot better than I do, probably, there is an unmet need in both hepatocellular carcinoma and cholangiocarcinoma, which is the development of minimally invasive diagnostic tools that can help with early detection, screening of at-risk patients, and disease monitoring. The development of such tools, diagnostic and minimally invasive tools, relies mainly on two aspects. The identification of suitable biomarkers, which can be accessed easily with minimally invasive procedures, such as a blood draw or a urine sample, along with the development of rapid assays that are suitable for point-of-care use, rapid point-of-care use. In the case of HCC and CCA, we already have proteins that are clinically used and paired with traditional diagnostic methods, as they are not specific and sensitive enough to be used on their own as indicators of disease. We are talking, for example, for CA19-9 for CCA or AFP for HCC. In the last decades, the interest of clinical research has shifted also on the study of microRNAs, which are deregulated and elevated in a number of pathological processes, and it is thought that each disease has a panel of elevated microRNAs that are associated to it. MicroRNAs are incredibly stable in blood, so the hope is that by achieving a multiplexed detection, so a detection of multiple of these microRNAs at the same time, this detection could be used in early diagnosis, screening, and disease monitoring. However, detecting biomarkers with established technologies presents a few limitations. First of all, limited sensitivity. It is also difficult to probe more than a few biomarkers at the same time. And many of these established technologies are not suited for rapid point-of-care use. Moreover, it is difficult to probe biomarkers at the low concentrations typical of the early onset of a disease. Recent advancements in nanotechnology, of course, have brought advancements in biosensing. However, many of these techniques require heavy sample processing, purification, immobilization, labeling, which again makes them unsuited for widespread point-of-care use. A solution to these problems can be given by nanopore technology, which is a single molecule detection technique. It is almost label-free. It requires limited to no sample processing and requires low reagent volumes. But what are nanopores? As the name suggests, nanopores are small apertures in the nanometer scale that are embedded in a membrane. This membrane can be made up of a biological substance, so we are talking about pore-forming proteins embedded in a biological membrane, or solid state, so pores embedded, for example, in quartz membranes, quartz capillaries or silicon nitride membranes. And nanopore sensing is a label-free, high-throughput single-molecule detection, meaning that we can detect the passage of each molecule going through the nanopore. The working principle is quite simple. Charged molecules, such as DNA, are driven through the pore in a process called translocation by the application of an external bias. And when a molecule passes through the pore, it modulates the ionic current that flows through the pore itself. If we look at the diagram on the bottom left, all the spikes that you see represent the passage of a single DNA molecule passing through the pore. So the aim of my project was to develop a nanopore-based sensor for the detection of microRNA biomarkers associated to cholangiocarcinoma and hepatocellular carcinoma. It has been shown that nanopores can, in fact, detect biomarkers, for example, biological molecules in general. For example, a colleague of mine was able to detect two proteins simultaneously, both in controlled buffer solutions and in serum. And again, another colleague of mine was able to detect 3 microRNAs at the same time directly from serum by combining standard solid-state nanopores with optical detection. However, despite the merits of these techniques, it is quite complicated to detect more than a few biomarkers at the same time. So for this reason, my project, instead of focusing on solid-state nanopores, focused on using biological nanopores. As I mentioned earlier, biological nanopores are made of pore-forming proteins embedded in a membrane. Compared to their solid-state counterpart, they have a smaller pore size and higher structural reproducibility. For example, one of the most used pores, alpha-hemolysin, has a nanopore aperture of just 1.4 nanometers. Thanks to the efforts put in by the research community over the past 20 years, biological pores have been engineered and a motor protein has been added to the top of the pore, which allows to slow down the translocation of the DNA itself. And this allows for the current signal arising from each of the single nucleotides to be distinguished, meaning that long DNA strands can be sequenced. The sequencing capabilities of biological nanopores have been perfected by Oxford Nanopore Technologies, which have created and commercialized portable devices that allow for DNA sequencing. Just to give you an idea, at the core of an Oxford Nanopore device, we have a membrane array with biological nanopores that all work simultaneously. These pores are embedded on a chip and are inserted in a flow cell, which can be inserted into the MinION device, which is a small portable device that works just by connecting it to a computer with a USB cable. And when an experiment is performed, the current disruption is measured from all the working pores in parallel, and the DNA strands are sequenced. However, these devices work really well when it comes to sequencing long DNA strands. So to detect microRNAs, we had to come up with a sort of workaround. We designed molecular probes specific for the detection of these microRNAs. The probes that we designed are made up of 3 parts: an adapter, which is the same for all pores and basically helps the probe to go through the nanopores, a target binding region, which is a sequence complementary to the target that we want to detect, and a central region called the barcode, which is unique to each of the targets that we want to detect. During an experiment, we have all the probes passing through the pores at the same time, and the current coming from all the pores is recorded. If a probe is unbound, it passes through the pore unencumbered, and it gives rise to a typical step-like current signal, which is then used for sequencing. When the barcode probe is bound to its target, it forms a double-stranded part, which is too big to pass through the pore. So for the translocation to occur, the target needs to, let's say, unzip from the probe itself, and this is detected as a signal delay in the current signal of the translocation that I have highlighted in yellow. So to give you an idea of how an experiment would work, So we selected 29 different microRNAs, 15 of which are associated to hepatocellular carcinoma and 17 to cholangiocarcinoma. And we designed 29 different probes to detect each of these microRNAs. So each probe detects is specific to each microRNA, and each probe has a specific barcode associated to it. To give you an idea of how an experiment works, we mix the barcodes with a sample containing the microRNAs, and we let it incubate for about half an hour. We then record the current using the ONT devices, and then we detect each event corresponding to the passage of each of the barcodes through the pores. And from these events, we are able to base call them, so to retrieve the sequence associated to each barcode. And this sequencing is followed by an alignment, meaning that the sequences that we detect are aligned to the library of barcodes that we know are there. And this allows to retrieve the information for each of the barcode populations. And then from each of these populations, we perform a delay analysis. So we see how many of the events have this delay that is related to the presence of the microRNA. So to make it simple, the barcode tells us which microRNA we're looking at, and the amount of delay tells us if the microRNA is present or not. This is quick. It requires minimal sample processing and minimal sample volume, just 30 microliters. So we performed a series of validation experiments using synthetic DNA targets in controlled buffer solutions to make sure that the platform and the detection method was sound and reliable and reproducible. The first test that we did was to perform experiments with one barcode at a time in the presence and in the absence of its target to see if effectively the percentage of events with a delay would increase when the target was present. And as you can see from this plots that represent the results for selected barcodes, when the targets are present, the percentage of events with a delay increases significantly. And this was repeated for all the barcodes, showing that most of them, except for one, have a significant increase in percentage delay when the target is present. The next step was to see if we could detect all the targets with all the barcodes at the same time. And if we if we look at the overall percentage of events which have a delay, we see that it increases linearly as the concentration of the target increases. And then by performing the sequencing and alignment, we were able to retrieve the information for each of the barcodes, so to look at each of the single targets and obtain binding curves for each of the probes, which I have reported here separately. And as you can see, most of the probes show a good dynamic range. The percentage of delay increases proportionally with the increase of target concentration. And overall, we have a limit of detection of around 100 picomolar. We then tested if we could use these calibration curves to estimate the concentration of unknown targets. So we mixed our barcodes with DNA targets at unknown concentrations. We performed the experiments as usual, and we then got the percentage delay for each of the barcode populations. And then using the calibration curves that we had designed, we estimated the concentration of each target. And I have reported the results. Sorry, this is not working anymore. Echo. The results here, and as you can see, for most of the barcodes, we were able to estimate the concentration of its target within, with minimal error and within 1 order of magnitude of the true value. So, in terms of these validation experiments, we were able to develop a robust and versatile platform. We successfully detected increasing concentrations of synthetic DNA targets in a multiplexed manner. Most barcodes show a good dynamic range, and the calibration curves allow us to enable to estimate unknown concentrations with good accuracy. The next step was to optimize this platform for the use with human serum. And when we transitioned to real serum and clinical samples, that's when the problems started. First of all, we realized that we could not use serum directly in the flow cells, as the device would basically break. All the pores would get blocked, and we could not get enough what we call events to perform a statistically significant analysis. And this happened even when we diluted serum. For example, here I show that even with serum diluted 800 times, the amount of events that we get is so low compared to when we perform experiments in controlled buffer. So we had to, again, come up with a workaround and test different serum purification methodologies. And the aim of these methodologies was to still keep the procedure as simple and as quick as possible. First, we tried centrifuging the serum with a 50 kilodalton molecular weight cut-off spin filter column, and we thought, this is great. It's quick, it's simple, it's fast. However, when performing spiking experiments, we were not able to detect the microRNAs that we had spiked in, so we had to abandon this idea. We then tested microRNA extraction, and we were able to detect spiked-in microRNAs, but only at concentrations higher than 100 nanomolar, which we deemed unsuitable for clinical purposes. And the last methodology that we tried combines albumin depletion, which removes the biggest protein present in blood, with a proteinase K digestion to help prevent the digestion and degradation of microRNAs by DNase and RNase enzymes. And the spiked-in microRNAs were detected successfully, so we decided to stick with this purification methodology. The final step was to test the developed platform with actual clinical samples. We tested it on 18 serum samples. 8 of them came from healthy participants, 4 from HCC, and 6 from CCA patients. And the double aim of these experiments was to check if we could actually detect microRNAs, and 2, if we could uncover any differences between healthy participants and cancer patients. So here I have reported the overall percentage of delays for all the patients and samples that we tested, and we expected to see an increase in percentage delay for the HCC and CCA patients compared to the healthy ones. However, as you can see, no particular trend is detected. We tried then to aggregate the results over all the healthy patients, all the HCC and all the CCA, and while the medians seem to slightly increase for the cancer patients, the populations are widely overlapping. So again, no significant trend can be detected. We then looked at the results for each of the probes individually, and we hoped that by looking at each probe and each microRNA individually, we could uncover some differences. But sadly, once again, no significant trend was detected. It was more on a case-to-case basis rather than differences between healthy and cancer patients. I was then curious to see if we could come up with strategies to classify our patients. So we performed a student t-test to determine which microRNAs were elevated, which I have reported in this heat map. And again, as you can see, no significant trend was detected. But again, I was like, let's see if we can use this to classify our patients. So I said, okay, what if we say that if more than 10 microRNAs are elevated, we classify the sample as a cancer patient? So I did that for the samples we had tested, and as you can see, only 10 out of 18 samples were classified correctly, given a platform sensitivity of 50% and a specificity of 62.5%, which is well below what has been achieved in literature by other types of testing methodologies using microRNAs. So, to conclude, can microRNAs be detected? Yes, they were detected. However, where no difference was highlighted between healthy participants and cancer patients. And this could be due mainly to 2 reasons: technological limitations or the choice of microRNA targets. In terms of technological limitations, it could be that our platform is not sensitive enough. Maybe the differences in microRNA levels are there, but we cannot detect them. So we should improve the limit of detection of the platform. Another problem could be that we lose too many microRNAs during the cleanup steps. These losses should be quantified, and if necessary, the protocol should be changed. And in terms of microRNA targets, the microRNA presence should be confirmed by performing RT-qPCR experiments, and if necessary, the list of targets should be updated. And once all of this has been optimized, many more samples should be tested. So, to conclude, the aim of my project was to develop a nanopore-based sensor to detect microRNAs associated to cholangiocarcinoma and hepatocellular carcinoma. I tried doing this by using barcoded molecular probes in combination with nanopore sequencing devices. We were able to build a robust and reliable platform, which is portable and versatile and works well detecting short nucleic acids in controlled buffer solutions. However, when transitioning to clinical samples, it gave inconclusive results. So technological improvements, as I said, are definitely needed. But I do believe that if these technological improvements are actually achieved, such a portable and quick platform could lay the foundations for a potential tool which could be used, hopefully, in clinical settings. So to conclude, I would like to thank my supervisors for the guidance and the help they've given me throughout this long PhD journey. I would also like to mention two of my colleagues, which I worked closely with, all of the EDEL group, and in particular, I would also like to mention the AMMF for funding part of this project and for inviting me to speak here. Thank you.